Okay, hello everyone. Thank you for joining us today for this webinar. My name is Valery Shkuropat. I am responsible for marketing and public relations at Fluigent, Germany. Let me please introduce our speaker for today. It is Daniel Kraus, PhD student at the Leibniz Institute of Photonic Technology in Vienna and also conference manager at SpectraNet. He will be presenting the topic microfluidic concepts and challenges for the characterization and sorting of bioparticles. Just a few notes before we get started. If you have any questions during the presentations, please use the chat panel available from the top right side of the screen. Our team will answer them right away if it is possible, or we will bring them up after the presentation. Also, the recorded version of this webinar will be available from tomorrow. And the presentation you will see right now will be also available for you as PDF to download. Now we will wait for a few minutes so the latecomers could join us and we could set up the presentation for you. Okay, good. Now it is time to start. Daniel, I give you the lead.
Okay, hello to my presentation. I will short introduce the IPHT in Jena and also the SpectraNet collaboration platform. And in the next few sentences, I will introduce myself. My name is Daniel Kraus. I'm studied pharmaceutical biotechnology at the Leibniz University in Jena. And also, I am a conference manager at SpectraNet. First of all, a few facts about the city of Jena. Oh, sorry. I can't see the... Sorry for the inconvenience. I have a little bit problems with the technical side. Okay, I will introduce the city of Jena, and we have in Jena around 111,000 inhabitants with 22,000 students with around 5,000 of scientists, 2 universities and 12 non-university research institutes. We also very good in the field of research. Many people in Jena work in research and development intensive manufacturers companies, and we have more than 350,000 350 patents per 100,000 inhabitants. The average in Germany are around 59 patents. We have also famous companies here located in Jena, for example, Zeiss and Jenoptik, and also the Schott company. And here you can see the skyline also from the nice city of Jena. I will introduce with a few words the Leibniz Institute of Photonic Technologies. We are with the slogan Photonics for Lives, from ideas to instruments. That means we build, or we research with photonics and photonic methods for diagnostics with focus on the photonic detection of pathogens, cells, bacteria, and all these small things. We also, by side, involved in the research of the coronavirus. For example, we manufacture the IR sensors for the ventilators in our clean room. And we also involved in the development of an antibody test against the coronavirus. The institute have three main topics: the biophotonics. We are located in the biophotonics with the bioanalytical and chip systems, and also with the microscopy and the imaging. And we have also the research unit of the photonic detection. That means the multi-scale spectroscopy and the ultra-sensitive detection. And we have the fiber optics with the nanoplasmonics and the fiber optics itself. But all these research units are very close connected to each other in the institute. And I will go over to my second workplace. It's the SpectroNet Innovation Cluster. And SpectroNet is a cluster with focus on photonics and machine vision. With more than 50 cluster partners, we are mainly focused in Germany and Europe. We connect companies, research institutes, and universities to accelerate developments as well as products in the area of photonics and machine vision. Here you can see the partners of the cluster are well-known companies from all over Europe. And with I will show you the three main activities of the cluster. We have the services, the conferences, and the project management. We offer our partners a range of different services. The services package includes, for example, the job portal, the media library with more than 2,500 videos of scientific and industrial presentations. And also we have an own academy for training, and we organize 3 conferences per year with a mode of 10-minute pitch presentations to present products, services, applications, or requests. This is a good opportunity to go in contact with new customers, potential partners, with a lot of technology like optics, camera technology, spectroscopy, illuminations, also funding opportunities. And if you are interested, I will be happy to provide you further information about the next conferences. And we are also involved in the initiation on support of innovation projects, research projects. We have experiences in regional and national fundings programs, and we have the contact to the supporting agencies. Here you can see a selection of our recent conferences. You can find all this information about these conferences on spectronet.de. So now I will start actually with the content today. I will show what we do in the microfluidics group at the Department of Nanobiophotronics, and I will also go a little bit more in the application way with my PhD topic and my work on the image-based flow cytometry and the spectral characterization and sorting of the bioparticles. I want to say in advance, I just want to give an overview of our activities. For more detailed questions and answers of individual topics, you have to ask also my colleagues from the group. But what you can see here is the different particles that we have available at the IPHT. The Nanobiophotronics group has many years experience in synthesis of nanoparticles. The spectral properties of the nanoparticles depends on their size and their shape, and also of the material which are the particles are made. In the IPHT, we can make spheres, prisms, cubes, stars, rods in a wide spectral range from 200 nanometers in the UV spectra to the NIR spectra range of the wavelengths. And on the right side, you see the nanoparticles diluted in water. You can see the different colors of the synthesized nanoparticles. We synthesize the particles in microfluidic chips because they have a few advantages in comparison with the classical batch synthesis. We have a higher surface to value ratio. Here you can see the microfluidic chip integrated in the cartridge. We need a lower consumption of materials. We can mix it much faster than the traditional batch because we have short ways or short channels. We can change the conditions very fast because the short ways, and we can control the temperature. And it is also possible to measure the particles online with to couple a spectrometer with a light source, and then you can directly in the channel measure the synthesis particles. We have also available the digital PCR in our lab. Here you can see the general workflow for the digital PCR. We have the sample aliquot in a droplet. Split this DNA, you have only one DNA per droplet. Then we can amplify the DNA, and at the end we get the result, an amplification yes or no in the droplet. We use this chip for the droplet production. We have 4 inlets: the PCR buffer, the sample, the PCR probes, and also the oil for the droplet generation. We collect all the droplets in an epi. The DNA is amplified with a conversion thermocycler, and after the amplification, the droplets are loaded into the readout chip. This is this chip here, and in this chamber here, we can image or we storage all the droplets, and then we can make the image-based readout. For this, we use the MFCS controller from Fluigent. It is also possible to use other devices like the PX module or the Flow-Easy modules, but we use actually the MFCS for the droplet generation. The droplets contours are hard to detect here on the right side. You can see it's a little bit blurry in these images, but you can see the positives and the negatives ones. You get some artifacts from the illumination, and also the vignetting is a problem. Also, when we change the filter from the bright field to the fluorescence, we get a little spatial error, and that we have to manage in the software. At the end, we have for each reaction vessel or droplet, we have a digital result, amplification, yes or no. By counting a large number of reaction vessels, about 30,000, droplets, we get a statistical significance of the process. The proportion of the reaction vessel is of the completed amplification is proportional to the amount of the DNA used in the amplified DNA sequence. So, and with our image-based readout, we can aligned the fluorescence image with the bright field image here. And at the end, we get a positive and a negative separated plot, and then we can read out the final result. Now to another topic, we have also available the droplet generation itself with dosing some liquids. And this junction is a T-junction with an integrated aperture. The droplet reaches as soon as the interface reaches the opposite side of the channel wall. The droplet volume can be exactly predicted from the geometrical parameters. And the droplet generation rate can be predicted from volume flow and the droplet diameter. For that experiments, we use the Flow Easy and also the PX OEM modules, and you see it works very nice. Here you can see it a little bit more in detail. When we increase the oil phase here, we get a bigger distance between the two droplets. So, and when we also speed up the water phase, we get a higher generation rate of the droplets. So we can play with also with the oil phase or with the water phase, and we can also play with the dosing unit. With this dosing unit we can easily synchronize dosing in the droplets. It is also possible to have a second or a third one of these dosing units. We can also add whatever we want to the droplets. With this chip, we can also create droplets with variable compositions. This is done by different dosing gradients. The mixing is enhanced by the winding channels here. For the mixing, you can see, okay, the mixing is very fast of the droplets. We have the uniform transportation velocity over the time, only the same distance between two droplets, and also with the same size, very uniform. So, and in each position of the readout loop, we have a different time that are the droplet in the chip. So we can easily find an optimum position for the readout. So we can read out maybe here, or we can also read out the droplets on this position. And yeah, we have the free choice for that. And also we can separate the phases from each other. That means when we are the experiment or the process is done, we can merge the droplets, or we can separate the oil from the water phase, and this is done by this capillary channels. The surface of these channels are wettable for oil and non-wettable for water. The capillary forces the inflow of the oil here in these channels, and the water phase is in the main channels. So with that we can control, or we can merge the droplets. We can also control the droplets by a self-droplet merging. That means we So that means the droplets waiting for the fusion partner. This is done by this bypass here. The droplet is waiting on the position of the another one, and after fusion, they goes straight the channel. We can also split droplets in the channels. So that we have droplets splitted with the same volume and the same size that we can use for analytical and also for reference or control sample. So we can search this in the channels or in chambers, and yeah, we can, after reaction with bacteria or cells, whatever we want, we can search afterwards one reference sample and one analytical sample. And when we bring all these functional stuff together, you have the alternating droplet generation, also done by a bypass. We can increase or decrease the distance between two droplets. We have the self-merging unit here. We can also generate droplets from here, mixing with the other ones from this side. And we can bring all this together in one chip. So we can also increase the speed in a manner that we want. It's not a problem. We have also an image-based sorting of droplets available. All these droplets passing the region of interest. In this region, all the droplets are recognized. And we have the decision point here. And when the droplet reaches the decision point, we get an impulse from the left or from the right side. Here you can see the impulse to sort the droplets to the left or to the right side. I would go a little bit in the next slides, go a little bit more in detail. This is only an overview that we can also sort droplets. Now I want to go a little bit more in the application way. So that means we have a current project and at the IPHT now with analyzing of honey. When you want to sell honey in a supermarket in Germany, you have to certify and classify your honey before you want to sell it. You have three main steps for the classification, but the most important or the most interesting step for us is the microscopic analysis of the pollen grains. So that means, or what we can see here is this is all the pollen, the honey samples of the last 3 months for certification. And this company is specialized in the origin and quality certification of honeys from all over the world. And this company can analyze 50 to 120 samples per day from the different honey samples from all over the world. The state of the art for the analyzing of this pollen is you have to count more than 500 pollen with a microscopic slide. You have to with a grid, and then you have to count by hand one by one the different pollen types. And at the end, you get a distribution of the different pollen types with a different percentage of the pollen. And when you have different pollen pollen, they are not really typical for the region or for this country. This is a sign for cheating honey, and this is not allowed to certify the honey, or you can't sell it in Germany. The state of the art is you have different work stages with bright field microscopes, and you have experts, and this expert know more than 2,000 different pollen types. And he individually focusing on every pollen grain in the set level to get which type of pollen it is. This is how the pollen looks like. They are very different in their morphology and also in the size, but it's quite hard to optical distinguish different pollen types from grasses because they are very similar. These types are quite different, but the grasses are very similar. That's the reason why actually the classification is only for that grass pollen. We use a microfluidic chip, the imaging flow cytometry chip. In the first, there are 4 special units. The first is to bring all this pollen together in the middle of the channel. Then we have a special rotation unit to bring all the pollen in the same focal plane. I will explain it a little bit more in detail later. And also do the second focusing unit to bring all the particles in the middle. And then we have the detection channel where all the particles are imaged. So here you can see the workflow. We have our region of interest and the pollen grains passing all in the same focal plane, the microfluidic channel. So for this, we have 3 main challenges. We have the motion blur of the objects. And this is the reason why we need very fast exposure times. So when we have a velocity of 1 millimeter per second, we need exposure times 1 millisecond or below. And this is why we need a lot of light and a fast camera to make sure that we don't have the motion blur. We have the problem of the depth of focus. The channels are have a height of more than 200 microns, and our objects have a size of around 20 microns. And with a 20x microscope objectives, you have typically depth of focus of around 1 micron. So, and also we need a high resolution because we have very small objects. To get the feature from the surface, we need a high resolution of the objects. And here you can see the different or the one pollen type at different points of view. So it's very hard to classify the pollen when you have only one image of the pollen. So they are looking very different. When you look at this images here and this image, you have very different morphologies from different angles. This is the reason why we use an image series and let the pollen grains rotate in the channel. And over the series, you see, okay, the pollen grains rotate, and with the angles shifting step by step, you have a 3D image, and you stack all pollens together, then you have a 3D image of the pollen grain. So the rotation is done by the off-center position of the pollen grains. You have a parabolic profile in the channel. It's the same like in river. In the middle of the river, you have the highest velocity, and to the walls of the river, you have the velocity near to 0. And when we bring the pollen grain in an off-center position, you have here a higher velocity than here, and the pollen grains turn on to rotate. For the classification, we use a convolution neural network. We have 15 different classes at the moment available from different pollen types, and we bring all these classes for training in the neural network. We use a pre-trained neural network VGG16, and we only train the last 3 layers for our network. And at the end, we have like this. This is the raw image of the camera. We normalize the image and we set a threshold and recognize all the different pollen types and the different pollen. Every pollen get an ID and a prediction of a class. So we have the class 1 to 15, and we have for each pollen we have an ID here. So we want to look at this pollen grain here with the ID 71. In the next image is also is tracked. We have the same ID, and we have also is predicted as class 4. In this image, the neural network says, okay, you have now class 2 because it's a little bit more mutated. Okay, the network is not so sure that it's in class 2. Okay. In the next image, the network says, okay, it's class 4, and then you see also class 4, and so on. And over the average of the single images, you can classify this pollen grain. With a single image, we have a false prediction, and over the series, we can be sure that the result class 4 is right. So at the end, we get a mean prediction. The pollen grain is classified as one class, except one time. And over the mean prediction, over 90%, we can say, okay, it is an hazel pollen in this case. Here you can see also the rotation of the sunflower pollen. And with this image series, we use the TomVis software to reconstruct the 3D information of the pollen grains. So we can make visible sometimes inner structures, or the surface of the different pollen types. But actually, our main focus is to classify the pollen. And then I will go over to another application, the imaging flow cytometry in combination with sorting. Actually, it's the same principle. We have the same units. In addition, we have also now sorting unit. That means we have here chambers filled with fluids, covered by an PDMS membrane, and with piezo stems we can actuate and sort to the left or to the right side. This is the optical visible area for the what we can image, and the other parts are covered with the connectors and integrated in the cartridge. So I go now a little bit more in detail. So we have at the beginning randomly distributed particles. So from the channel, all in different focal planes. You can see it here. Sometimes it's a little bit blurry, and some of the pollens are sharply. And with the focusing unit 1, we bring these particles all in the same plane in a vertical lamella. With the special rotation unit, we step with small angles this vertical lamella to a horizontal lamella. And then we have all the particles in the same focal plane. So all are sharply imaged, or can be sharply imaged. So here you can see that all the particles are in the same focal plane. And with the second focusing unit, we bring all these particles in the same position of the channel. So we can, that's important for sorting. So, and we can also do this here. You can see they rotate in the channels, and we can also have a 3D image of the pollen. We can sort by different parameters. That means we can also sort by diameter. We can sort by area, velocity, gray value, also granularity, some different moments of the pollen grains. I think we have more than 60 parameters available for the classification, and we can also combine different parameters with each other. So we can also with the area in combination with the gray value is also possible. And I want to go to the sorter principle, and that means we have the fluid chambers covered by the PDMS membrane. We have the illumination with the LED, and we have the microscope objectives on the other side. So, and with the particles are imaged, recognized, calculated, and at the right moment, we get a fluid impulse for sorting or for non-sorting. And yeah, the stem bring a little bit pressure on the chamber, and with this pressure, you can sort it to the left or to the right side. You can see here. So this is how the software looks like. We defined at first a region of interest. In this region, the particles are detected and calculated for the sorting. This is decision point. On this point, we have the impulse of the fluids from this side or from this side. And yeah, at the end, we need also the channel direction for it's important to, that the software knows in which direction all the particles flow. And at the particles, it's only one particle pass the region of interest, get an impulse at the right moment, and deflects in this channel. And without sorting impulse, the particle goes in this channel. This is in real time. I hope you can see it. The actuated particles goes in the lower channel, the non-actuated in the upper channel, and when they are too big, they want the software sort them out. So a little bit slower in slow motion. You see, this is a duplet of the. This is a duplet of the two particles. It's too big, and this is why they sort them out. Okay. And at the end, I will show you an outlook of what you can expect in the next months from us. We have a red stained pollen grain on the right side, and they pass a bandpass filter. Is directly in front of the camera, and here in the spectra you can see of the stain, and in the red area of the here in the red area you can see only the phase objects of the pollen grains. So and then it passes to the green, and the red stain absorbs the light, and then you see it's a little bit darker. So, and with that we get a full spectra inflow from the particles. This is also possible for cells, for algae, or whatever have spectral, or we can discriminate spectral properties. So, and at the end, we want to sort by their spectral properties, but it's a little bit more in future, and with that we have to do some work. And at the end, I will thank my colleagues from the microfluidic groups and also the guys from Fluigent to provide me all these devices for my research, and for the trustful work together. And with this, I want to thank for your attention, and I'm open for questions.
Okay, Daniel, thank you very much for your presentation. Now it is Q&A session. So please. Daniel, you can see the questions on your left. It is marked for you. We do not hear you right now. Daniel, we still do not hear you.
Oh, sorry.
Yeah.
Yeah. The problem is with the fixed image of the class slide. We have the problem.
Daniel, one moment. Could you please read the question so we could understand?
Okay. Yes. Okay. So sorry. Yes. Why not just simply applying software for the image recognition to a fixed image in the class slide? Why you are using microfluidics point grains for microfluidics for point grain classification? The advantage is we can analyze much more particles in a short time. That means we can analyze hundreds of pollen grains per second. So an expert needs on a microscopic slide needs to classify them manually pollen by pollen. They need a lot of time because they have to focus in the set position to classify pollen grain. With the rotation and the tracking of the pollen grains in the microfluidics, we can analyze more hundreds of pollen grains at the same time. So we can analyze in 1 minute some thousands of the pollen grains. So and we have a better statistics of the report, and also we can store all the images if we want for manually classification by a human. It's also possible. And when you analyze the slide, you bring it to the bin, and then it's done. So you can't look later on your data. So we can storage all to the protocols, all this data if you want. So I hope this is the answer of your question. And can I go to the next question? I think so. Okay. And the second questions.
Yes, please.
Have you tried to develop a platform for more than one kind of particle? Yes, of course. We as a microfluidics group, we only develop actually microfluidic structures. So we tested our microfluidic structures with real samples. So and we use the pollen particles for the classification or to figure out how it works, because we have a lot available in our lab. We can also the pollen are very stable, non-toxic, and we don't have problems with growing or contaminations or something like this. And they always looking at same. When you have blood cells or something like this, every cell it differs a little bit. So when you have hazel pollen, and the hazel pollen looks every time as a hazel pollen, so this is very easy to detect them or to classify them, and to make sure that our system works well or right in this case. Okay, I go to the next questions. For the focusing of the pollen, why you don't use a very flat microfluidic channel? Yeah, that's also a good question. We have the problem that in the honey we have a lot of fossils and also material or bigger material in there that we have to filter or to keep sure that the microfluidic channel is not blocked by this. Sometimes you have also 10 or 15 or maybe some more particles. and they block the microfluidic channel, and then you have a problem. So the process is broken or is stopped by the blocking of your channel. When you use bigger sizes of your channels, in my case it's around 250 microns, you have no such problems with clusters of the particles, maybe clusters, and also with rods or something like this. When you have this in the sorting area or in the rotation area, the rotation it does not work anymore. So, and then you have the problem. With this, we can take care of that. We can also use it when we have some problems with the clusters of pollen and something like this. The honey is a nature product, so you have a lot of different things in there, all from the bees and the insects. They bring this materials inside the sample, and that's the problem. Why we don't use smaller or flat microfluidic channels. So then we have another question: why would we want to have a continuous droplet stream versus continuous droplet stream? I can't. What what means this question? Maybe you can help me.
The droplet stream versus continuous stream. You see the difference? Daniel? So the question was about the continuous droplet stream versus continuous stream.
I think about it. The droplet has the advantage that you have in reaction chamber, a small reaction chamber. So when you encapsulate droplets, you have the digital PCR. So you can also sort the droplets at the end if you want. It's an idea. So you have the droplets in your hand. So, and you can also merge the droplets together, and you can also work afterwards with your bacteria or with your encapsulated yeast cells or whatever you want. So when you have it in the continuous stream, you can also do this for an assay or something like this. But sometimes when you only want to have the positive, or when you want to grow yeast in the droplet and it's fulfilled with yeast, and then you have some empty droplets, you can also sort them, and then you can process your sorted yeast cells that are really encapsulated in a defined vessel. So actually, this is the difference. So it's a little bit depends on your it depends on your application. So there's I hope this question is with that answered. We have another question. Can microfluidic devices filter out the different types of the pollen impurities? Yes, it can, but we have the problem. We have an unknown. Often we have an unknown sample. That means we don't know what is in the sample. So when we filter it, we have the problem that maybe one fraction, one size of the pollen, maybe bigger ones, the more than we use a filter of 50 microns, and we have pollen with 60 or 100 or 200 microns. We filter a complete fraction of the sample. So this is actually why we can't filter it so much, because we have to care about that we have all the pollen, different pollen types in the sample or in the microfluidic chip. So and with that, when we use bigger channels and bigger filter mesh, we are safe that we have all the different types in the sample. So it is also, when you have further questions, please let me know, write me an email. We can also discuss in B2B, or when you are not happy with the questions. Which technology do you use for the fabrication of your microfluidic structures? We have an own clean room in our institute. We manufacture these chips, or the microfluidic chips and structures, by wet etching with photo lithography. And we have some experts in the groups that make the simulations and also the cut images of the structures, and then we produce it in our clean room, and we use mainly bioparticle glass chips. So we also have, I think, polycarbonate chips available, but actually we use mainly for research. We refresh it and then we use it again. We use the glass chips, so we don't have so much. We don't need so much different chips for our research. So actually, that's okay. So then we have another question. For the self-controlled merging, what is the mechanism of the droplets waiting, and how fast it could to be merged for two droplets? For that, I will. For that, I will go backwards. So, okay. Yeah, this is the. I think you mean this. The mechanism is when the droplet is in this position, the flow goes over to this section here and speeds and waiting, and this is the mechanism why they're waiting for each other. So we can also see it in the. You can also see it here. It's a little bit slower. So the droplet blocks this position, and the flows goes over to this, and with that you bring, or you pulse the droplet in this direction. I don't plan this structure. If you need more information about these structures, please write me an email. I give you the contact from the designer of this structure, and he can explain it a little bit more in detail, I think. So, and I think the question, and another question, what throughput can be achieved? Yeah, it depends on the flow rates. When we speed up the flow rates of the setup, you can produce a lot of droplets, but we have to test it. But I think you can produce a lot of droplets with that, and also with the microfluidic analysis of the point grains. When we speed up the flow rates, it depends on the hardware. So when we use better hardware with more processing power, we can also speed up the system. So, but I think we go out of time, so I think we are finished. And yeah, please send me, if you have further questions, please send me an email. So, and I will ask as good as I can. So, thank you for your attention, and have a nice day.
So thank you, Daniel. We really have some more questions. We will try to send you an email with answer on it. But if we want, you can also always send us your questions to the contact at fluigent.com. We want to thank you for participation. We hope you enjoyed it. We remind you that the recording of this webinar will be available from tomorrow. So you can always check our website and the platform. And there is a dedicated page with a list of Fluigent's upcoming webinars and at our website. Also, if you have any wishes or ideas for new webinars, also please write us. At this point, we wish you a pleasant evening, pleasant rest, and bye.